Welcome, everyone, to our latest installment of the Spark Seminar Series. Dr. Lee is the professor and chair of the Department of Molecular and Human Genetics at Baylor College of Medicine. His research focuses on developmental, translational, and clinical studies of skeletal dysplasias and inborn errors of metabolism. Today, he will discuss with us the translational pipeline that his group has developed for designing novel gene therapeutic strategies to reduce osteoarthritis pain and disease progression. And he'll tell us about the clinical trials to test their safety and efficacy in patients with OA. He's a member of the REJOIN Consortium, which stands for the Restoring Joint Health and Function to Reduce Pain. That spells out REJOIN. It's part of the Helping to End Addiction Long-Term HEAL initiative, which aims to elucidate how joint pain mediating neurons innervate different articular and periarticular tissues. The research groups are working together to generate harmonized understanding of the neurobiology of the pain pathology relationships in humans, gaining valuable insights for comparison with preclinical models. All data, models, protocols, and tools generated across the REJOIN teams will be available on the Spark portal alongside over 350 curated data sets, models, and simulations from multiple organs, species, and research groups. Spark ensures interoperability across diverse contributions to advance basic and clinical applications, as well as bioelectronic devices. Additionally, Spark connects the body and the brain with open resources that support research into systems physiology and neural control in the periphery, such as our computational platform, OSPARC, and the annotated maps which are generated from SCAN, our knowledge base of peripheral connectivity. Spark is a member of the AWS Open Data Sponsorship Program, which enables all public data sets to be stored and accessed without cost to users and contributors. The Spark portal shares complex multimodal data, meeting NIH, HEAL, and journal standards for scientific data repositories. Speaking of journals, we are very excited to communicate that we have partnered with Autonomic Neuroscience, Basic and Clinical. By integrating data deposition into the publishing process, we're advancing our missions to make research more transparent and impactful. If you're considering submitting to ANBC, we hope that you'll consider submitting your data and protocols fairly to Spark. We're here to support you with grant language for data sharing plans, and we also offer services related to computational modeling and simulations that can be utilized to augment your research, as well as support the incorporation of non-animal models into your research. You can scan that QR code in the bottom for more information if you're interested. Before I turn it over to Dr. Lee, however, I just want to alert you to our next seminar in 2 weeks. On September 17th, Dr. John Osborne will delve into interoception and provide an overview of the role of the afferent renal nerves in regulation of renal function and development of pathophysiologies, both renal and non-renal. Be sure to join us. We are building out the schedule for the next Spark seminar series, so if you are interested in presenting or would like to nominate someone, please take a moment to fill out that survey at the beginning of the Q&A session of today's talk. Questions are welcome. Please pop them in at any time during the presentation using the Q&A panel in your Zoom window. We'll have time at the end of the session for Dr. Lee to reflect on your questions and comments. The seminar is being recorded and will be available on the Spark YouTube channel soon after. And with that, I turn it over to you, Dr. Lee.
Thank you, Susan. I'm delighted to represent the REJOIN Consortium here and also to participate in this Spark seminar series. I'm delighted to talk to you about a topic that we've been working on for really almost 2 decades, and that is developing gene therapy approaches to treat the complications of osteoarthritis. These are my disclosures. I think the one that's most relevant here is we have licensed our gene therapy for osteoarthritis to Pacira Biosciences, and they are leading the clinical validation studies, which I will also describe. So I don't need to tell this audience that osteoarthritis are always a major health issue in the US, over 30 million adults, enormous economic burden. And 1 in 4 adults with broadly all forms of arthritis, of course, experience severe joint pain, greater than 7 out of 10 in terms of pain scales. So it's a very significant burden on our society and a big focus, of course, of the studies in Rejoin as well as HEAL. We've been focused on delivering therapeutic genes using a virus-mediated approach. So as many of you may know, gene therapy has been something that has been in the development since the 90s, since when I started my career. And after all these decades, it is a reality. And there are both viral and non-viral approaches. In terms of localized delivery into the joint space, we have focused on viral mediated delivery primarily because of the challenge of transducing the tissues of the joint. I've studied connective tissue biology for all that period of time, and I can say that the matrix itself is an evolutionary developed barrier to invasion. And so really viral mediated approaches give us an advantage. And these are 3 of the main platforms that have achieved quite a bit of effectiveness in the gene therapy space. Adeno-associated virus, which is a small non-pathogenic virus. Adenovirus, which I will talk about, which is really one of the early platforms because of its efficiency in infection and gene expression. And retroviruses, oncoretroviruses, because of their ability to integrate. And this has primarily been the platform of choice in cells that are rapidly dividing because of their integrative nature. And this is, for example, in hematopoietic disorders. I'm going to talk primarily about our work with adenovirus over this past several decades. Now, when you think about just sort of the evolution of gene therapy clinically, there were several milestones. I think the 3 include the first ever clinical approval of a product in the world, and that occurred with a p53-based adenovirus delivery in cancer, and that was in China. A second was an AAV-based approach to treat a rare metabolic disorder, inborn error of metabolism, lipoprotein lipase deficiency in Europe. And then really in the US, the first ever in vivo gene therapy, and I had the opportunity to participate in the FDA advisory committee that made the recommendation on this therapy, which is, of course, the AAV-based gene therapy to treat an RPE65 mutation that caused a form of blindness or retinal dystrophy. So really, it's been, I would say, in the past 2 decades that clinical gene therapy has become a reality. And in fact, and there's probably 1 or 2 more than this, but at the beginning of this year, there were 43 FDA-approved products in this space. So continuing to evolve. Now, when you think about treatment for osteoarthritis and delivery into the joint, there are, and this is not unique for this application, but for many, we think about in vivo gene therapy, where the vector is directly delivered into the tissue, the example being the RPE65 gene therapy for blindness. Here, we're thinking about delivering directly into, for example, the intra-articular space, or into skeletal or muscular tissue directly. And of course, there's ex vivo gene therapy, where one transduces cells either with viral or non-viral vectors, and then reintroduces the cell back into the patient. And this is a good example of, for example, the sickle cell gene therapy that was approved at the end of 2023, and many of the hematopoietic targeted gene therapies. Again, we've been focused more on in vivo approaches. Now, the platform that I'll be talking about has been a development that occurred here from the late 90s and really focused on the safest version of the adenovirus platform. And this is something we call high capacity adenovirus. And it has other names over its evolution. It's been called helper dependent adenovirus, for example. One of the real strength of adenovirus is very high efficiency transduction. Sort of pound for pound, probably the most efficient vector for transducing non-replicating cells. One of the negatives is it can stimulate, especially when exposed to the immune system, a strong adaptive immune response. The early generation vectors, which were, of course, replication deficient by the deletion of early transactivators like E1, were developed and then subsequently modified for additional deletions that reduce the production of viral proteins, hence increasing their cloning capacity and reducing their immunogenicity. What we at Baylor developed several decades ago was a version which was the least immunogenic, which was the deletion of basically all of the viral genes of adenovirus, thereby really eliminating late viral protein expression or leaky protein expression after transduction. And this reduces the adaptive immune response and facilitates long-term gene expression with many, many model systems, which I'll touch on. You do need in the production a helper virus, basically what would be an early generation, first generation virus, which produces all of the proteins needed to generate the high capacity ad with the transgene in trans, but then it itself could not be packaged, and I'll explain how that was developed. Importantly, the key to making this practical and ultimately, I think, clinically relevant is the production advances that allowed for really elimination of the helper virus and very low cost production of the vector, which is really something we, especially now that proof of principle for gene therapy can occur, we're very focused on in terms of actually getting the treatment to many people. So early adenovirus generations or vectors, basically, as I said, deleted these key transcriptional factors that made it replication deficient. These are key genes within the 38 kilobase double-stranded vector genome. So it has in theory, an enormous amount of space to carry a transgene. And then subsequently, second generation deleted multiple genes, which reduced the immunogenicity. What Phil Lang really did here at Baylor years ago was develop a system to produce a completely deleted vector. So basically all the viral genes are gone, replaced with human genomic DNA and the transgene of interest, flanked by the inverted terminal repeats and the packaging signal. Importantly, a producer cell, an E1 transcomplementing producer cell, is also infected with a helper virus, which is basically a first generation adenovirus vector expressing all the genes in green that allows the packaging of the high capacity ad. Importantly, the packaging signal is flanked by LockP sites, and this producer cell expresses Cre recombinase, which eliminates this packaging signal. Hence, only the high capacity ad is able to be produced, and the helper virus, which produces the proteins in trans to package the high capacity ad, is excluded from packaging. And in fact, simple 1 to 2 liter preps can make a ton of vector, over 10 to the 13 viral particles or genomes, and can be purified at high levels where the contamination of the helper virus is less than 0.01%. When tested for by a variety of methods. This is an example of a southern blot, for example, where basically you cannot see here in the prep any signal from the helper virus. So, and then further production changes now allows for large scale generation at very low cost, which is again a key advantage of the system when thinking about low cost gene therapy, something which is the goal for the future and the present. And I'm not going to go through all the data, but basically, unlike early generation adenoviruses, we and others have studied this over the years and showed that it can rescue a variety of clinical phenotypes in a variety of tissues. We focused on early on in the 2000s on metabolic diseases like OTC and mouse models, and you can see this long-term correction compared to early generation adenoviruses. Here's a mouse. This is studying another liver disease in the gun rat. Again, you can see dose-related correction. Hemophilia models in a dog, you can see very long-term correction. So again, unlike early generation vectors, which are eliminated due to the adaptive immune response, because of the absence of viral proteins being expressed, you get long-term expression. And then similarly in muscle, you can get long-term expression in a different series of different contexts. Now, the barrier to this vector, as we think the adaptive immune response becomes really much less of an issue, is still the innate immune response. And in fact, this is relevant for pretty much any gene therapy approach. AAVs, for example, can stimulate an innate immune response at a lower level than AD. But if given at high enough doses, you similarly can see a response. And we've seen that in clinical trials at very high doses of AAV. And the same with even non-viral vectors. And we actually very early on recognized this in the context of systemic delivery when the vector is exposed to the reticular endothelial system in the liver, effectively the largest innate immune organ in the body. And there is a dose responsive. One of the markers of this is a dose responsive thrombocytopenia. You can see platelet counts dropping, as well as transient elevation of liver enzymes. So this has now been repeatedly shown with a variety of different vectors if you go with a high enough dose. And again, you can see that even with helper dependent, you get the same response, which is not shocking because again, this is due to the innate immune response, which is a response to the vector, protein, and nucleic acid. So really, because of this reason, systemic delivery of these vectors are not clinically an option because of the innate immune response. And much of the field has moved to AAV in that context. But from the context of localized delivery, such as in the joint or other closed spaces, it's an excellent option. And this study really demonstrates in the context of intra-articular injection how early generation adenovirus with a vector expressing here luciferase, because of the adaptive immune response, is lost after a few weeks, but you can get lifelong expression in the joint with a dose of the high capacity or helper dependent ad, as it's called here. And in fact, when we compared it to different types of AAV in the joint, these were vectors, since we're not an AAV lab per se, generated by the National AAV Vector Corps when we did this study. You can see efficient transduction of synovial tissues. But again, high capacity ad has significant advantages in terms of the level of transduction. And this was validated independently by our collaborators in Pacira and previous to them in Flexion as well as Genequine. They use a LAC-Z vector and transduce knee joints. And you can see the degree of soft tissue in the knee joint that was transduced by this sort of open view. And so really, it was sort of the obvious next step to target a disease and a complex disease like osteoarthritis. We had been interested in studying genetic forms of OA, but obviously the common form of OA would be another target. And as we all know, there are no really disease-modifying therapies out there. The target that has been sort of been well described in at least in terms of clinical experience. So it's not actually yet been proven in terms of phase 3 studies to be efficacious, is blocking IL-1. And this was sort of post hoc data of the development or the study of IL-1 inhibitors in clinical trials, which showed that, again, post hoc, that there were really a decrease in time to total knee replacement and time to total hip replacement, suggesting that perhaps blocking IL-1 is an effective approach. And we know that there's tons of preclinical models that support this, but it's a challenge in terms of clinical efficacy for multiple reasons. The life of the duration of the protein, the challenges of maintaining a level that is relevant in the knee joint. And so we felt that a gene therapy approach potentially could be the best way to achieve this type of effect. So I'm not going to review this pathway. It's well studied, and I'm sure many of you are aware of it. But I would say that IL-1 signaling in the context of skeletal biology as a whole is very high up in driving many of the processes that are detrimental in the context of osteoarthritis and heterotopic ossification. Things like endochondral ossification and chondrocyte hypertrophy, elements like inflammation as it leads to the impact on the neuroimmune environment. And so this was very attractive. One important element was that in recombinant IL-1RA type trials, a natural inhibitor of IL-1, there's been no demonstrated long-term efficacy. And some of the gene therapy trials that have been proposed and done to date focused on constitutive expression. And while this has been shown to be safe and improves OA in animal models, as you'll see similarly from our preclinical studies, we took the different approach of really proposing to express IL-1RA from an NF-kappa B responsive promoter in the context of a high-capacity ad. And the reason we took this approach was the simple hypothesis that, local inflammation has been demonstrated to be important in regeneration of multiple tissues, but only when sustained causes injury. And so this regulated expression, which increasingly is going to be an important sort of principle in moving gene therapy forward, I think will be critical in terms of safety and efficacy. So we developed this vector and studied it in multiple surgical models of OA, the DMM model, as well as the cruciate ligament transection model. And we showed that by both structural measures as well as behavioral measures, here I show you the structural and histological measures, there was improvement just comparing red to green in many of these measures, supporting that there was an efficacy signal. What's key is, of course, how does it perform in large animal models, and often is a required driver in moving forward into human clinical trials. And here we took advantage of collaboration with the late Alan Nixon, using the equine, the horse, carpal osteochondral chip model. So again, a surgical model of induced OA in conjunction with exercising. And here we injected the same vector, an equine IL-1RA, driven by an NF-kappa B responsive promoter. And what you see is expression of the IL-1RA at multiple doses, as well as improvement in different functional measures. I would note that, and this is actually interesting because it presaged a clinical observation in the studies that I'll touch on, that we didn't see significant differences in the two doses which were used, a high and a low dose, which was one order of magnitude different. And just keep that in mind, but different certainly from placebo. And this is just one example I will show of one of the horses that was treated back then with placebo. And this is looking at range of motion. So you can see the limitation of range of motion. This is the carpal joint in which the injury was produced. This is at the end of the study with the vector, which again supports in this case the high capacity ad. So supporting really moving this vector forward in terms of clinical study. And that's in fact what initially Flexion with GQ and then now Pacira licensed the product from us to pursue. And so this is a phase 1 study as a single intra-articular injection of the product called PCRX-201. Basically, it's the identical vector with the human IL-1RA driven by an NF-kappa B promoter in the context of a high-capacity adenovirus. And it's now just released the 3-year data in moderate to severe neo-OA. So these are adults, 30 to 80 years, with moderate to severe OA, and it was widely inclusive of KL grades 2 to 4. So really all comers. Individuals had prior treatment failure of greater than 2 other therapies for neo-OA and a significant evidence of a baseline WOMAC score, pain score. And there were low, middle, and high doses of the vector given via ultrasound-guided intra-articular injection. And only acetaminophen was permitted as a rescue analgesic for the first year. Other treatments were permitted only at the investigator discretion after the first 60 days following treatment. Other analgesics like SSRIs and other agents like that were on stable doses if the patient were already on these treatments. Now, what was important is that both a steroid not pretreated as well as a steroid pretreated group was included, and for multiple reasons. Many of the gene therapy products and trials and over the past decades, and now pretty much most of the gene therapy treatments include a steroid injection for blunting the innate immune response. as many of you may know, steroids can have a positive effect on pain. So the trials that have been performed, show efficacies maybe out to as much as 3 months. Um, uh, but again, there are concerns long term because of potential chondrotoxic effects of repeated intra-articular steroids. So, in terms of the safety data, there are no serious treatment emergent AEs that were related to procedure. There were common AEs, specifically knee effusions, that were self-limited. They generally resolve within 1 month to 2 months. More common in the not steroid pretreated group, 61 versus 36%, and was not dose related. Now, what is very interesting, as I'll show you, is that pre-existing adenovirus neutralizing antibody did not affect the response, and I'll touch on that. What is really exciting is that after a single intra-articular dose, 3 years, and similar results were seen at 1 and 2 years, but these were the 3-year data that were just released and presented, is that PCRX 201 improves pain and stiffness in this group of patients. And least mean squares improved in the context of these WOMAC A and B scores, both in the not pretreated and in the pretreated group. So you see the pain WOMAC A score here, as well as the WOMAC B stiffness score. The least mean square means improved across the 3 doses, both relating to stiffness for WOMAC B, as well as in the not pretreated and more in the pretreated groups. And you can see that again here. It's interesting in the sense that, and this is where this correlated with what we observed in the equine experiment, and to some degree the preclinical rodent experiments, that there did not, at the doses used, seem to be a dose response to the effect. And this may relate to the fact that we were hitting a threshold dose and also the regulated gene expression that is occurring in these in the local environment. Also, looking at the KOOS subscore in terms of activities of daily living, the ADL, again, across all 3 doses, there was improvement in both the not pretreated group and the corticosteroid pretreated groups. And you can see that here in terms of the improvement. And currently, the phase 2 study is actively occurring. And this is a blinded study with an active steroid control. Most, as I said, it would be surprising to think the steroid component would mediate this effect for 3 years, given what we know about steroid effects. But it is certainly, I think, possible that it enhances the effect of the gene therapy, as we've seen now in many other types, including AAV gene therapies for multiple genetic diseases, where steroid treatment does blunt the immune response. So I think the immune response here is more the innate immune response than anything else. This is interesting because, of course, all of us generally have antibodies and neutralizing antibodies to adenovirus serotype 5, which is the vector that is commonly used. There does not seem, at least from the phase 1 data, to be any difference in the response of the WOMAC A and B scores to pre-existing baseline neutralizing antibody. And you can see that here. This is interesting also in the context that, of course, the joint may be a somewhat privileged site in terms of transduction. And we are still thinking about this in terms of other alternatives, in terms of if this were to become an issue, because I think repeated treatment, obviously, for a chronic disease would be indicated. What the steroids do is they seem to facilitate resolution of an induction of a neutralizing antibody. So it's not surprising that with injection, and we see this in the preclinical models, as you'll see, that there is an induction of neutralizing antibody against AD. And in the presence of steroids, there is a blunting of this and a resolution of this to a lower level, which again may facilitate re-injection at a later time point, given that our preclinical studies support that this modifies the disease course, but obviously, of course, doesn't prevent it. And over time, there will be continued progression, albeit delayed progression. So this is where, again, it's clear that given the complexity of OA, any monotherapy will be insufficient ultimately. And this is where, in thinking about genetic causes of OA as well as environmental, we sort of leaned on our experience to ask, well, what are alternative approaches and other targets? And this is where we focused on what human genetics has taught us. Rare human genetic conditions can point to important in vivo targets for drug development. And this is a very interesting genetic disease, a very rare human condition called camptodactyly arthropathy, Coxa-Vara pericarditis syndrome. Um, Focus on the arthropathy component for right now, but basically these are loss of function mutations in proteoglycan 4 or lubicin, which are secreted proteins from superficial zone chondrocytes as well as synovium. And when there's loss of mutations, you develop early onset OA, as well as some deformities like camptodactyly. And you can certainly see the pathological abnormality here. This is a section in the joint showing synovial hyperplasia. There are multiple splice isoforms in other tissues, and interestingly, patients develop a pericarditis. We hypothesized that this was a great target. Since loss of function causes early onset OA, we see this in preclinical models, we see it in patients, human patients with this rare condition. And so we asked, and this was a graduate student, Mario Juan, years ago, asked whether overexpressing PRG4 would protect from OA. And histologically, as well as by micro CT phase contrast imaging, we see protection. So here, for example, look at yellow, which is a treatment with the vector intraarticular in a transection. This here is a cruciate ligament transection model in the mouse. And this is long-term, again, quite effective. So if you look at these reconstructions of phase contrast scans of placebo on the left versus on the right, articular cartilage in a treated mouse joint, you can clearly see the preservation of cartilage. Subsequently, another grad student, Adrian Stone, with a postdoc role, asked a question, well, could we use a vector, two vectors, expressing PRG4 and IL-1RA in a combinatorial gene transfer? And basically, we saw what was supportive of a synergistic effect on both cartilage preservation and different measures of potentially pain, in this case, hyperalgesia, in this rodent model, control the two vectors and the combination. So this supported what is currently a trial in the equine model of, in fact, monotherapies of the two, benchmarking it to the existing IL-1RA approach, but also monotherapy with PRG4, as well as a single vector, again, the advantage of high capacity ad, expressing PRG4 and IL-1RA. And we expect this now multi-year trial to be completed by December. So we're excited to see the effects in the equine model on this approach. We're hopeful that it would be effective and perhaps provide, sort of a synergistic benefit. Ultimately, whether it's the same vector or different vectors, we do expect that serial injection will be needed. We're thinking about, a disease that evolves over 2, 3, maybe more decades. And so we began to look at the other advantage of high capacity ad, which is the switching of serotypes. There are multiple serotypes, and they can be easily switched in production purely by changing the nature of the first generation helper. So the vector doesn't change, the nature of a specific protein changes in the helper virus, which again, it can be made very easily at very large scale. And so Jensen Yang, a postdoc in the group, currently studied this idea of switching the serotypes and showed that with different serotypes, effectively transduce equivalently with some mild variation in the joint long term. And so this is a number of weeks, intra-articular injection. You see a very typical profile. In all cases, there is a rapid loss and then stabilization. And this we have studied for many years to know is related to the innate immune response in terms of both silencing of certain vectors and clearing of a certain initial amount of cells, but then there's long-term stable expression. And in fact, as a proof of principle, if we were to make the most robust, and so one could, in the mouse model, you can make a very strongly immunogenic model for anti-AD by intramuscular injection of AD. And then following cruciate ligament transduction, do multiple injections of different serotypes expressing either therapeutic transgene or a marker. In this case, we did a LAC-Z marker. And what you see is, not surprisingly, with intramuscular injection, we can get a very strong anti-AD response. And then if you then re-inject with different serotypes, you can get minimal transduction with the same AD5, but more transduction with the other serotypes, 1, 2, and 5, similar in efficacy to a naive mouse, for example. And not surprisingly, if you actually look at vector genomes, you would see a similar correlation of vector genomes within the joint. And this type of injection, switching of serotype, can enhance transgene expression in the context of repeated. And you can see here an example of LAC-Z staining as well as genomes within the joints. So we think that whether it's the same serotype, because we see that that is certainly possible, or better yet, switching of serotypes long term, one would have a viable strategy for moving this forward. So we believe that eventually this type of combinatorial gene therapy will be important for long-term targeting of OA. And it may be with approaches like IL-1RA, which is showing enormous sort of hope right now in clinical trials. In fact, the FDA gave the product RMAT designation last year based on the phase 1 studies and the results from that. Or it could be with approaches like PRG4, which has been known to impact as a lubricant biomechanical stress in the joint. Though I would say it is likely PRG4 also has an anti-inflammatory effect, given not only the human genetic data that patients with loss of function have inflammation, for example, pericarditis, but we do see in our preclinical study impact in this signal. So we do think that this type of combinatorial approach will be important long term. I think as a whole, this platform has advantages. While it cannot be used for systemic delivery, but for localized delivery, there are significant advantages. If one's thinking about large genes or multiple genes or regulated gene expression, this platform can be used. So we're collaborating with multiple members of Rejoin to extend it to their models, like in the TMJ, as well as other OA models. This is critical, and I've become especially passionate about that. While many gene therapies, which are amazing as successes, the cost of the product is prohibitive. One advantage of this platform is it can be produced at very low cost. And as an example, millions of individuals have gotten adenovirus injections. If you've had a COVID vaccine with the J&J product, in fact, that is a serotype of adenovirus. It can hold other types of machinery that are requiring multi-component, like prime and base editing, and even large-scale genome editing could be considered, and not necessarily relevant for OA, but in terms of other applications because of the space. So it's in this context that we're now trying to understand how specifically therapies like this impact the pain. Why do we see, for example, in the human trial, this very dramatic reduction in WOMAC-A pain scores? And similarly in some of the preclinical models. And as was introduced, the Rejoin Consortium really focuses on understanding the neural multi-omic input into osteoarthritic pain. And these are some of the examples of the preclinical models, including human, that are being studied. And this sort of multi-model approach is, I think, really powerful and key in understanding mechanisms which are preserved, effect sizes which can translate. I think similar to what we did in the preclinical rodent, equine, and then now human by Becerra, I think really underscores the power of this approach. And as was also described, this is a wonderful consortium of multiple institutions. We are at Baylor College of Medicine and our KNERV, and we run the administrative core. We work closely with the data center, and all of the data eventually will be made available on the Spark platform that are generated, and we're part of course, the larger HEAL consortium. And just as a highlight of what we're doing and the data that are being generated, we're generating multiple animal models of OA, which I've touched on. Ben Arenquio's group at Baylor is looking at viral tracing, including retrograde tracing from the joint. Russell Ray is trying to do genetic tracing and focus on the sympathetic nervous system, while Josh is focused at UVA is focused on tissue clearing and 3D imaging, and Ray Chen is focused on multi-omic analysis, especially spatial transcriptomics, and Neela leads the administrative core for the whole consortium. And really, the key for us is identifying how aging, exercise, and gene therapy impacts pain and mediate, signals from the joint as it travels to the DRG, as well as potentially to sympathetic ganglia. And I think the whole consortium has been focused on building a set of toolboxes to label DRG sensory and autonomic joint innervation in multiple models of OA, including TMJ in the knee, clearing methods and tracing methods to directly visualize changes in this context. And ultimately, which is the deliverable mechanistic targets, which can facilitate our understanding of these types of processes and intervention, and also perhaps potentially identify new targets. From our group, Russell and Abdinello, his postdoctoral fellow, has really done a systematic analysis showing that for sensory neurons, genetic tracing with NaV1.8 Cre, and importantly, in a new area, using the DBH Cre lines for tracing sympathetic neurons, we can actually see clearly those that are innervated and labeled from the joint. And these are examples of this genetic tracer with TD tomato co-labeling with a neuronal marker like TUJ1 in both the knee synovium, in the DRG here on the left, as well as in the sympathetic ganglion, as you see in the merged signals for the multiple markers. So really encouraging, and also combining it with chemical tracing, comparing different types of chemical tracing directly from the joint into, for example, the DRG. Similarly, effective ways to clear a very difficult tissue like the joint is important. And Josh Wythe, as well as our group, have compared both organic and aqueous-based approaches to clear the mouse knee joint effectively, while labeling with the vasculature with lectin-based fluorescent markers, and now using on top of that IHC with neuronal markers to see again changes with all the different variables which I had mentioned before. And then finally, I think the alterable sort of deliverable will be looking at spatial transcriptomics here in the DRG, but led by Ray Chen's group, but also in the trigeminal ganglion in the context of TMJ and dysfunction. Building a spatial transcriptomic atlas here in the DRG that could identify the subset of DRG neurons, which, for example, would be changed or improved and or other non-neuronal cells, as I think we're beginning to see in the context of NEOA and hopefully in the context of treatment with gene therapy. So with that, I'll end. There are many, many folks. I'm honored to speak on their behalf, both within our gene therapy subgroup and the rejoins group of my lab, the Becerra group that is leading the clinical trials, our equine collaborator, Heidi Ressing, previously the late Alan Nixon, and many of the folks within the Rejoin Consortium, both in our KNERV group as well as the broader consortium in terms of collaboration. These are the picture of members in the lab, and here specifically are our KNERV Rejoin team I've touched on and I've mentioned, and our funding over the decades. Thank you, everybody, for your attention.
That was spectacular, Dr. Lee. Thank you so much for walking us through the rich research plan that you have been carrying through over the past few years. We have a number of questions already submitted. I invite others to drop any questions or comments into the Q&A panel, and we'll open it up for your feedback. Additionally, you'll see that a survey just popped up. If you have an idea for a topic or a presenter, or you'd like to be one, please self-nominate. Let us know, and we'll get in touch. So, Dr. Lee, we have a couple of questions. One is, I'll start off with this one. It says, in your earlier slides, you showed autoantibodies did not affect effectiveness. Were autoantibodies measured in the blood or the synovial fluid?
Right. So the study, so I think you're alluding to the study, the phase 1 study from the Pacira trial. Those were baseline neutralizing antibodies measured from the blood. And not surprisingly, individuals across all the doses, individuals with all the different severities of OA had pre-existing anti-adenovirus 5 neutralizing antibodies. I'm sure all of us here have it. And they did not seem to impact the WOMAC pain scores. It's interesting. We have also done the study. There's no question that in the mouse model, since this was brought up, the mouse model may or may not predict what happens in the human. That's not shocking. In the intramuscular immunization, which I view as the most robust model for blocking antibodies, we can block transduction in the joint with pre-existing. But when we have done the experiment with serial injection, while we do seem to attenuate transduction, we have a signal, and I did not show that because it's still preliminary, that suggests that even with serial injection with the same serotype, even though transduction in this most robust context is inhibited, we may get a prolonged efficacy signal compared to a single injection. So we don't understand that, and I didn't really want to show that until we have more stronger data. But it does raise the question, and I think when you see these signals both in human and in rodent models or preclinical models, it does raise the question that perhaps the joint is somewhat immunoprivileged to some degree. So we'll see how that works. And again, you do see some slight trends with the minus versus plus steroid. So that again could be another element in this. But we do believe that the joint is a somewhat privileged space immunologically to neutralizing ad.
That gets to another question that was posed actually, and this one was posed earlier, so it was perhaps relevant to this initial introduction about inflammation and the viral vector delivery. And they wanted to know whether or not you could supplement the vector delivery with dexamethasone to help suppress inflation.
Yeah,
and so
well,
how does that factor into that immune privileged, locale that you're talking
yeah.
about? The
So
joint.
in fact, as I said, I think that's absolutely correct. That will happen. That's happening in all gene therapy vector platforms, right? AAV, for example, which is we now have multiple experiences in vivo delivery. Steroids are given. Now they're given systemically with prednisone often. And the reason is AAV is given systemically. The immune response to AAV is different than the immune response to AD. Interestingly, and this was seen with the very early clinical trials, they evolve over several weeks. And in fact, the toxicity can evolve over that period of time. It's very different than AD. And I think AD's component is more of a typical innate immune response. And this is why, in fact, the methylprednisolone or the depomedrol was given in these phase 1 clinical trials. And going forward, I think that's going to be the approach that will be used. As I said, in the phase 2 trials right now that Pacira is doing, they have an active steroid-only control arm, which is important to do, obviously, to exclude formally that this is all a steroid effect. I don't think anyone believes that, given what trials have been done on pain and steroids for it to last 3 years. But I think it has to be, formally proven.
Yeah. So speaking of that clinical trial, there's a question about the PCRX 201
201.
clinical trial. And wanted to know if you saw a difference in the KL2 versus the KL4 patients.
So just as a disclaimer, I'm not part of that trial because of conflict of interest, obviously. But from the public data that I'm aware of and what's been presented, what the company has presented to me, there was no difference in terms of response. But again, keep in mind, these are still small numbers, right? It's one of the largest, it is the largest OA clinical trial ever done in gene therapy, but this is where, again, the phase 2 will be important. I think in the phase 2, what the design is that, again, KL4 will be included, but they are capping the number of KL4 in the phase 2 study.
That's really cool. So so before we shift gears to a little bit more into your current and future studies, there's one more question about the data that you've previously provided. And actually, I'm going to combine two questions because I think they're highly related, but uh, you can disabuse me of that if it doesn't make quite sense. So the IL-1 levels are often low in OA, which does help explain the small drug effects in the previous trials. So there's two questions. Why did you choose IL-1R over TGF beta 1? And then also, have you tried to overexpress IL-1B to show an increase in pain? So, sort of the flip side of things, to show an increase in pain and other OA symptoms. So that's the mashup between the two questions.
Yeah. So we have not done that experiment. Um, you know, so the answer is no. So I have no information on that that's, informed. I think with TGF beta, there are obviously currently there are products focused on TGF beta. I have studied TGF beta in many, many contexts in skeletal diseases. And the challenge with TGF beta is somewhat akin to why we put IL-1 RA behind an NF-kappa B promoter, right? It gets at more the broader, I would say, scientific and perhaps, somewhat philosophical approach of targeting signaling in general. I think that, when you have extreme autoimmune inflammatory condition, and they're both, genetic and non-genetic examples of that, then, you know, sustained blocking of that pathway is how you get a therapeutic effect. I think when you're modulating these types of much more chronic conditions that I would say where different signaling pathways contribute to the ultimate pathogenesis, i.e., there's complex pathology, as that slide showed. Not sure that, well, what I would say is my experience has been, and there's a lot of literature and data I can point to, and we don't have time for it, that there's an optimal window. It's kind of the statement, too much and too little are both bad. And I think
Mhm.
TGF beta is a good example of that. We, in fact, developed an anti-TGF beta clinical trial product for brittle bone disease, which is another part of what we work on. And, I think that in the skeleton, especially and cartilage specifically, we've studied this issue where excessive TGF beta and too little TGF beta can both have unwanted effects on cartilage biology. And we know that also in the context of inflammation, right? TGF beta effects are context dependent. Depending upon what's around it and how much, you can both pro and anti-inflammatory. And in one context, it could generate, tolerogenic, dendritic cells, and in the other context, it can stimulate strong Th17 cells. So again, I think this is where again, not saying it wouldn't work. It's just I've stayed away from that context in terms of, especially in a complex disease like OA, what is the right amount? So it's challenging, I think, that in targeting signaling pathways like that. This is, again, part of the rationale for the regulated expression.
Mhm. So looking towards the research that is being undertaken by the Rejoin Consortia, There's a question about the value of spatial transcriptomics. And so they want to know, what does it offer to dive in spatially? So to perform spatial transcriptomics for the DRG analysis. And wouldn't a singular or a single nuclear RNA-seq be more realistic for a ganglia? I'm not
So
sure.
that's a great question because it's something I think our consortium has been grappling with now for 2.5, 3 years. So we're doing both. So we're doing both single nuclear RNA-seq as well as bulk, as well as spatial. I think they all have their pluses and minuses, obviously. Um, Spatial, by definition, if given sufficient resolution, can give us direct assessment of cell interaction, sort of paracellular signaling events. Um, Now, that's only useful if we have sufficient sensitivity to identify what's changed, right? I mean, I think whenever I look at new technologies like this, will it tell me more than a simple H&E stain? And I think the hypothesis is that by giving higher resolution, we get more information. I think no one disagrees with that, especially in terms of cell-to-cell interaction. What I think is key is whether we can get enough data to identify the key targets beyond saying this is this type of neuron, right? Which we could have done with targeted IHC or what have you. So it's going to be that issue of how much can we push sensitivity, because what is the potential deliverable will be, and this is where single nuke would may or single cell or single nuke could give us insight into, but it's all going to be pushing on the technology front. Can we get enough sequencing to identify that low expressed target that really drives the phenotype? And I think the answer is unknown. I think we're all, it's going to depend on technology. The technology is evolving. What is available right now commercially, there's no question there is sort of a ceiling on what can be seen. So
Mhm.
it's, can we push beyond that ceiling?
Yeah, and I think that's also where how nicely the consortia as a whole has leaned into the team science approach and really worked hard at harmonizing your aims, your goals, your methods, and your implementations to try to create that generous pool of data across as many loci as possible. So as many places as you're looking, by unifying where you can, how you collect that data, it helps provide a little bit more power.
Absolutely. I think that's the key, right? It's the rigor component,
Mhm.
which is so important in terms of preclinical and clinical studies. But as I hope our work and our approach has shown, or at least pointed to, the signals that are most likely to translate, and I have worked in the preclinical and human space for a long time, and often when you think about translation, it's usually, preclinical models tell you what can happen. They don't tell you what will happen. But as you see the signal replicated more and more in different contexts, in the TMJ, in the knee, in a mouse, in a rat, in a horse, that's going to more likely predict translation in the human context, because what is the human context but a completely outbred population, right? That's why clinical trials are so difficult in the OA space. So I do think you're absolutely correct. It's only by these types of multi-site collaborations that we see the replication of those signals.
Yeah. And just to, as we wrap up, just to throw out another source of potential integration and opportunity is that on the Spark portal itself, we incorporate data from many research labs, not just your own team science group that's working on rejoin, but we've got contributors from the Precision Human Pain group that are contributing DRG sequencing data at a heavy clip, as
And
well
let me
as
just
data.
interrupt that.
Yeah,
We have taken full advantage of that. That data, which is great data that they've generated, has informed our analysis. So I
that's
think that
awesome.
exactly.
Yeah, and then there's also looking at that sympathetic nervous system side of things that your group is also looking at, like bringing it to the research that was undertaken by the Spark program, which was focused entirely on the autonomic nervous system, having all of that data hopefully together and harmonized using similar principles is hopefully providing a good playground for people to like find and recombine data in new and exciting ways. So
Absolutely.
I'm thrilled. I really appreciate your time, Dr. Lee. I appreciate everybody who's joined us today, and I really hope that this gets you excited for the future of potential therapies that may be coming soon to help all of us who are getting older and potentially experiencing that way.
It was a pleasure. Take care.
And take care, everybody.